Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. So today we will discuss about the microRNA and what is the microRNA and related to the gene silencing mechanism. So the microRNA and their gene suppression or silencing of the gene process we will discuss. And microRNA is used for gene silencing. For example, cancer cells. Cancer cell is produced due to the activation of that gene which produce as cancer. So the oncogene is present in inactivation due to microRNA function. So the microRNA used for the inactivation of the cancer gene, oncogene. So here is the general mechanism to produce protein is the functional DNA which that contain genes, and gene is transcribed by the produce a messenger RNA is known as the transcription, while this messenger RNA go into the cytoplasm to produce translation by the different mechanism to produce protein, and it means the gene activation. That's produced in the form of gene activation. So we can say that gene is active, while here is a person which that contain a different type of cell. So let's talk about the half we can take a tissue to understand the gene. So here is the gene tissue contain a cell, and this cell contain nucleus, and here is the nucleus. Nucleus contain a chromosome. For example, human contain a pair of the chromosome, which that means the one pair is the 26 chromosome in the form of pair 23. So chromosome contain histone and DNA. DNA is the double helical structure, which that is one strand is going to 3 to 5 prime, while the other is going to 5 to 3 prime. It means the phosphodiester bond which that is occur with the glucose molecule. So here is the different region contain a gene. This gene is the specific for the production of specific protein. So here is the transcription factor which that attach the gene to site or gene something to transcription factor to promote transcription. It means the polymerase enzyme used to bind and produce the messenger RNA. This messenger RNA is inhibited if it is harmful our body. So it is inhibited by the microRNA. So here is the region which that is used for the microRNA gene, which that transcribed, and microRNA which that is inhibition of protein synthesis from the messenger RNA. So that is the general overview of the microRNA silencing of the gene. So this mechanism is like that. So remember. So let's talk about here is the, we can say that here is the different type of cell contain different type of same type of nucleus, while it means the gene of activation in the same region, while here is off and on. So the off and on of the gene silence or express of the gene in the same chromosome. So the DNA is the same in our body, while its gene switch off and on to regulate gene activation in specific areas. For example, muscle, liver, bone, eyes. For example, muscle express that gene, but in the liver it will be not expressed, but it's going to off. So that means during the off or on, it's depend on the region of the cell. So here is the different type of cell will be produced in our body due to the off silencing of the gene or expressing of the gene by the translation. So let's talk about the gene dominance. What is the meaning of gene dominance? Gene dominance, for example, here is the cell which contain nucleus, and nucleus contain 46 chromosome, while we are understanding that, for example, it's plant cell, so contain a different type of nucleus in the form of pair, and one chromosome, homologous chromosome, or homologous chromosome which that is in the form of acetylation and methylation. For example, E, D, C, B, A and A region contain activation of the chromosomal gene to produce protein, while on the rest of the E smaller d is the suppression of the gene due to the dominance on the smaller letter on the capital letter is the dominant. So here is the general structure that gene silence due to the dominance of the gene on the other homologous chromosome. So that means here is, for example, DNA, which that is present in the chromosome, and the DNA polymerase bind and transcriptional factor will promote the transcription. So that transcription means the messenger RNA will be formed. This messenger RNA produce the translation of gene into protein. So the next, I hope you make sense about the gene dominance. Next here we will discuss about the gene silencing. So the gene silencing, a basic concept of gene silencing of the DNA is known as epigenetics, and epigenetics play a role after fertilization to the whole life. It means during the fertilization obviously is the genetic, while the rest on the rest of the development and growth is depend on the epigenetic. So here is the DNA, which that is microRNA genes contain, while here is the DNA, and the cancer causing gene will be activated and thus produce the messenger RNA, and it is inhibited by the microRNA by the production of the same region where the microRNA gene will be produced, while the breakdown of RNA, a messenger RNA, will be formed and no protein will be occur. So epigenetics mean during the fertilization of sperm with the egg to produce zygote. So from fertilization to form a zygote is the genetic. It means that every animal use this mechanism, while on the other hand the zygote will be developed into the embryo, and the whole organism is depend on the genetics by the methylation and acetylation on the requirement of the environment. So it means the requirement on the basis of environment that gene will be occur, and if the methylation and acetylation will be occur. For example, I give you example. So the methylation will be occur in the liver cell production. So it means that all gene of the muscle cell producing gene and bone cell producing gene or some other gene will be silenced due to the methylation. So this is the methylation mechanism, while the acetylation will be occur for the reliable or comfortable for that protein synthesis according to the liver cell, for example. So it means the gene off and on the basis of requirement of the related to the environment is due to the epigenetics, and epigenetics play important role in our body. Without epigenetics it's impossible to live longer. On the other hand, the epigenetic is important for the survival of the cell. For example, skin cell will not convert into the other cell, so remember about that. So next here we will discuss about the mechanism of microRNA to silencing of the gene. So here is the nucleus, which contain a chromosome. This chromosome is made up of DNA and histone protein. So the DNA of the different region contain here. Here is the microRNA gene, which that produce the microRNA. Remember about that. While here the transcription factor, helicase enzyme, primase, and promoter to bind to stimulate the RNA polymerase to produce after the unwinding to produce RNA. This is the messenger RNA. This will express the oncogene. For example, it is produce a cancer causing gene, which that express to produce protein to rapid proliferation of the cell. So remember, so this messenger RNA will go outside through the nuclear pore, and it means if it will be go, so it will be translate into harmful protein. So here is the microRNA, which that is transcribed into the primary microRNA, which contain intron and exon. Exon is the basically not useful for the making a microRNA, so it is splicing by DROSA or DGCR8 is the enzyme, which that is using for the cutting of the exon. So exon will become a messenger RNA. This messenger RNA will be go into the outside of the from the nucleus to the cytoplasm, and this is the good and functional for that. So that means it is used for the functional protein synthesis, rather than that messenger RNA will be produce translational protein is the harmful, for example the cancerous protein. So it means the intron will be left behind by the drosha cutting, and that go into the smaller precursor microRNA that is still primary. So the exportin will be exportin 5 protein will be bind to export this microRNA into the cytoplasm. So there are many mechanisms into the cytoplasm after the releasing of exportin, that is the AGO1 complex, and that AGO1 complex will be formed, and here is a tail and loop containing microRNA. So the dicer is attached with the AGO1, as well as the TRBP is the intermediate binding sequence. So it means the AGO1 is bind. It means here is the microRNA which that is cutting by the dicer. So the terminal loop will be separate after the cutting. So the left is the double-stranded microRNA will be occur. So the AGO2 loading will be formed. It means AGO2 loading will be attached. That microRNA, double-stranded microRNA, will be separated into the single-stranded microRNA. So these single-stranded microRNA are present. One strand will be released into the cytoplasm, while the one is in the form of AGO3 loading, and that means the risk is the complex. It is known as the RNA induced silencing complex. So it means the R risk mean the RISC is the risk is the RNA induced silencing complex. That means the induced silencing complex will be silence the messenger RNA, which that is the harmful, and produce the protein which that is the harmful, and silent it or stop. So our breakdown of the messenger RNA, degradation of messenger RNA, or the silencing of the messenger RNA by the binding of that risk RNA induced silencing complex. So here is the RNA, messenger RNA will be bind with the RNA induced silencing complex and form a double-stranded, and that means here the transfer RNA, and transfer RNA used for the protein synthesis, which that pick the amino acid from the cytoplasm and carry toward that go into the ribosome, and ribosome will be three A site, E site, and P site. P site is the middle basically, and A site, P site, and E site, exit site, and amino acyl site is the form, and that means it will be stop and no transfer RNA come again. So it means the polypeptide will be not terminate, and it means that stop the mechanism to produce to translation. While here the breakdown of messenger RNA is also occur by the splicing by the binding of these microRNA to splice or cut. So thanks for watching. Please subscribe my channel and watch another video and support me, give the suggestion.